Hi, Michelle Glass here. Welcome back to another episode of our Lecture 6, Chapter 6 series. And our topic for this video is fermentation, one of my favorite processes. This process makes yummy bread and cheese and wine and beer and lots of other important products for us, right? So fermentation is marked as being an anaerobic process that is producing ATP in the cell. And it's based on what we've already learned about glycolysis. So glycolysis, we know, is an anaerobic process. This is where we're going to be breaking down our 6-carbon glucose molecule into 2 3-carbon pyruvate molecules. As a part of this, we are oxidizing glucose and reducing NAD plus into NADH. We see that NADH is going to take those electrons to the mitochondria, deliver those electrons to the electron transport system, make more ATP. And as a result of that process, we're going to recycle, remake NAD plus. So that's all great. That's what we've already been learning about in our aerobic respiration, right? So what happens if there is no oxygen available for the mitochondria? Remember, the mitochondria requires oxygen. So if there's no oxygen required, how are you going to get your NAD plus recycled back into the cell for use? And so that's where fermentation comes into play. There are 2 different methods of fermentation. So the first we'll talk about is lactic acid fermentation. We do this in our skeletal muscle cells when our muscle is working to its peak level of activity, so that might be super fast or really holding something, using that muscle to hold a load for a long period of time. Lactic acid is another one of my favorite types of fermentation because this is used also in the making of yogurt and some cheeses. So I like dairy, if you haven't picked up on that. Alrighty, so what we see happening in this case is, of course, glycolysis, as we've already studied, but then notice that pyruvic acid molecules are going to be converted into lactate molecules, hence the name lactic acid. In order to oxidize those pyruvate molecules into lactate, we, I said it wrong. In order to reduce those glucose, in order to reduce those pyruvate molecules into lactic acid molecules, we will see that our NADH is going to bring those electrons to that process, and that's going to recycle our NAD+. Okay, so now I think I've got it right. We are oxidizing NADH, making more NAD+, and we are reducing that pyruvate as we're turning it into the lactate. Okay, so again, the key thing with fermentation is we need to somehow return the NAD+, that little taxi cab, back to glycolysis so that we can keep making ATP in that glycolysis reaction. Our second method of fermentation is alcohol fermentation. So of course this is going to be producing beer and wine, and also involved in bread making and some other processes here. So what we see happening again is we need to have this method of remaking that NAD plus at the end, and we can do this here because our pyruvate in this case gets changed from a 3-carbon pyruvate to a 2-carbon ethanol molecule. So in addition to allowing us to replace our NAD+, in addition to making the ethanol, we're also making carbon dioxide gas here as a waste product. So that would be the bubbles that you get in your beer, or that's giving us the bubbles that we need to make bread rise. We are again reducing the pyruvate so that we can oxidize the NADH back to the NAD+. So that's the basic pattern that you see in both types of fermentation. The pyruvate is reduced, turned into something else, and this then oxidizes the NADH back to the NAD+, because we're not able to do that at the mitochondria. Okay? That is fermentation. That's it for now.